Thank you so much for the invitation to this very special conference. It really fits well with the interests of my lab, which are in sialic acid biology, human evolution, and uniquely human diseases. So I wish I had the time and energy to join this wonderful group of people who are moving this whole field forward, but I haven't. But just to assure you that I do care, even though I think globally, I have acted locally. So what happened is a few years ago, about 8 years ago, I got upset about the fact that there was no teaching of evolution in medical school. So I tried Harvey Feinberg's, when you hit B on the screen, it goes black, and everybody wonders what happened. It's a way to get attention. So I took Harvey Feinberg's frontal assault role and I went to the administration and said, I want to teach evolution. I got rejected outright. So then I decided to take what he would call his guerrilla approach and the camel's nose under the tent and found somebody teaching genetics and said, give me an hour. So they gave me an hour. And of course, I talked about human evolution. The kids went wild. They said, how come we don't hear more about this? So I got established in one corner of the tent. And then I got a little bolder a few years ago, and I said, well, you know, there was another friend of mine became in charge of the introductory course, and I said, these kids are coming from many different backgrounds to learn many different things, but they learn to know about one species, humans. So the very first lecture they should get is, what is a human? And for the last few years, I've been successful in doing that. And so I actually give lecture number 2, which is actually lecture number 1. The first one's an orientation about understanding human evolution. And of course, the kids are bright-eyed and bushy-tailed and ready to learn. So, but that's as far as I've gotten. The camel is not yet in the tent, but got partway in. Okay, so, and if you're interested, I've written a little article based on this lecture, if it helps you in any of your efforts to do the same. So let's return to my topic at hand, which is disease difference between humans and other hominids. Starting with the outstanding first lecture of this symposium, you've heard a lot about how it's so important to compare humans with other animals, and One Health and veterinary medicine and public health combined all together, and I'm very much in favor of that. But I'd also like to suggest that one should consider the possibility that major diseases of a given species are likely to be related to maladaptations during the recent evolutionary past of that species, and therefore the comparisons of disease incidence and susceptibility between humans and our closest evolutionary relatives should be useful. And of course, I don't really have to introduce this figure, which shows the relationships between the so-called great apes and us, and our close relationship to the chimpanzee, hominids, and hominins. So where would you make the comparison? Unfortunately, unlike the case with many monkeys, the numbers are very, very, very small. And just about the only group you can make a comparison with is captive chimpanzees, a few thousand that have been cared for in captivity, mostly in the United States. But these captive chimpanzees have had excellent medical care, better than many Americans, and also get full autopsies and so on. So I think they're about as comparable as they can to make a comparison with humans. And so I went to the Yerkes Primate Center and other centers and tried to learn more and more about diseases in apes. And I was told that the commonest cause of death was heart disease, heart attacks and heart failure. I said, oh, at least in that way, they're just like humans, and I forgot all about it. And my spouse and collaborator, Ajit Varki, who was a pathologist, went back and said, what are you talking about? It's a completely different disease. So it turns out heart disease is common in humans and chimpanzees, but is caused by a completely different pathological process. On the left side, you see humans, and there's a typical atheroma about to blow open and cause coronary thrombosis, the typical heart attack of humans, or the ischemic heart disease that results in heart failure. On the right side, you see the chimpanzee that dies of a heart attack due to an arrhythmia. It's caused by massive fibrosis, interstitial myocardial fibrosis, shown with that Masson chromosome staining. So there are actually two mysteries which are unsolved. Why do humans not often suffer from this fibrotic disease so common? By the way, this has been reported in orangutans and gorillas and bonobos. And conversely, why do great apes not often get the kind of heart disease that we get? So I think here's an example of where the comparative approach with our closest relatives is very useful. And this is not due to differences in risk factors. Besides the very high cholesterol that chimpanzees have, they have several other risk factors, which I've listed here. And they should be having high rates of atherosclerotic coronary thrombosis if they were like humans. But one can really not be sure about these things with this sort of level of comparison. So a few years ago, Nissi and I tried to summarize everything we could find out in collaboration with a few chimpanzee veterinarians and divided up diseases into diseases where there seem to be definite, probable, and possible differences. And among the definite include coronary thrombosis and this interstitial myocardial fibrosis, falciparum malaria, bacterial sexually transmitted diseases, typhoid, cholera, AIDS, etc. And the others that are less clear, like hepatitis complications, Alzheimer's pathology, carcinomas, and the other ones which are just possible based on lack of any reports. But there's never been a case so far I found of bronchial asthma in a chimpanzee. Okay, so for the rest of my talk, I'm going to focus in on what got me into all this, which is a study of sialic acids on cell surface and secreted molecules. Every cell in your body is covered with a dense and complex array of glycans. It's what I call the dark matter of the biological universe that biologists have yet to tackle. We are now working with the standard model of the biological universe. This is at concentrations ranging from 10 to 100 millimolar on the surface of your cell. And these terminal sugars are found in various types of glycoproteins and glycolipids and secreted molecules like the oligosaccharides you heard about. And they're found primarily in animals of the deuterostome lineage, and interestingly, on the pathogens that associate and attack deuterostomes. So we got interested in this first from my background in oncology because there are two major kinds of sialic acids in mammalian cells, nu5AC and nu5GC, and they differ by a single oxygen atom. AC is the precursor of GC, so it's converted to GC. Excuse me. It was claimed that this was missing in humans, but present in cancers, but that humans had antibodies against it. And to cut a very long story short, many of you have heard this story. We basically found that while all the species had the precursor sialic acid nu5AC, we were missing nu5GC due to a mutation that occurred about 2 to 3 million years ago in our lineage, resulting in a single event that's fixed in the human population. So we're like knockout mice. We basically have a deletion of a single gene, accumulation of the precursor. And we've looked at many consequences of this, but I'll just talk about a couple of them. One of them that may have something to do with how this was first selected is falciparum malaria. Some of you probably know the killer malaria of humans is a very, very recent disease that emerged in the last few tens of thousands of years and was probably occurred with a single transfer from one ape into humans back from a classic form of ape malaria. And in some years ago with Pascal Gagnon, we looked at this question and basically found that the falciparum malaria merozoite likes to bind AC and the ape malaria likes to bind GC. And so we made a hypothesis, which Pascal and I then extended in this article. And the idea is that we hominins once suffered from the same type of malaria that apes suffered. And at some point, there may have been a selection and loss of the GC target for this or other reasons, and we got a free ride for a while. But the parasites always win in the end, right? And so one particular strain of the single jump occurred where it learned how to bind the nu5AC sialic acid. This is consistent with all the facts we have to date, yet to be fully proven. So more recently, another example that was just recently published in the host adaptation in Salmonella typhi. Now, typhoid fever is a human-specific disease. Large doses of Salmonella in the days when this was possible to do were given to chimpanzees, and they really didn't get very sick. And in fact, the typhoid bacillus didn't proliferate very well. And the exotoxin of typhoid, which is really the cause of typhoid fever, it makes it much worse than most Salmonella, didn't make the chimpanzees very ill. And to cut a very long story short, these days we have glycan arrays where we have different sugars sitting on the edge of sugar chains. And you can see on the left side that the typhoid toxin binds wonderfully to the human sialic acid and simply cannot recognize the ape sialic acid that has only one oxygen atom difference. And using some model mice, we further went on with this, including some crystal structure work by Jorge Galan, and showed that we can show this typhoid fever sensitivity and resistance may have been resulted from, of course, the salmonella evolving towards humans. So then with carcinomas, we've written a recent review which is in which we conclude that we cannot be absolutely certain about differences in carcinomas, but it does appear to date there's not been a single epithelial carcinoma in any great ape reported. Could be ascertainment bias, but we're not entirely sure. But there is one thing that we can be sure of, and that unlike all other carnivores, humans are very susceptible to red meat-induced carcinomas. And the red meat is associated with a variety of diseases. And to cut another very long story short, we found that we are eating this non-human sialic acid. It's getting incorporated into our tissues as a xeno-autoantigen. We're making xeno-autoantibodies against which we react, getting a kind of inflammation called xenosialitis. And we can now show that this combination can cause inflammation both in epithelial cells and endothelial cells, perhaps aggravating carcinomas and atherosclerosis. We now have a very good mouse model where we've shown this actually happening. We have to prove it formally in humans. Now you would think that we now know the costs of risk, and public health has been telling us what to do all along. And I don't know if you believe in this concept of microlife, but this one concludes that one burger is 30 minutes of your life. So you would think that at least the people in my lab would have got the message. No, this is a picture that they sent me. All of them eating ribs. So I would point out that I think evolution has left us with something which is the biggest challenge that this society faces. And that is outlined in this book that I co-wrote with my recent deceased co-author, Danny Brower, about our incredible ability to deny reality. And so I think this is the biggest challenge we face in this field of public health. So having found one sialic acid difference between humans and apes, we got interested in this pathway in which there are many different sialic acids involved and many different types of sialic acid binding proteins. And I'll have to skip through this for lack of time, but basically what we found is that these sialic acid binding proteins that recognize sialic acids act as self-associated molecular patterns and recognize sialic acids as self, as associated with danger and pathogen-associated molecular patterns. Now, what we have is bacteria that have reinvented sialic acid as wolves in sheep's clothing. They're taking full advantage of us, and there's a whole variety of them taking advantage of sialic acid reinventions by convergent evolution and attacking us, and many of them are human-specific pathogens. So we have many, many other stories of this kind, almost a dozen sialic acid-specific changes in human evolution. But I'll just close with one last one, in which we found that Siglec-6, this is messenger RNA expression, it's massively, massively expressed in the human placenta. And this expression is uniquely human in the human cytotrophoblast, not present in ape placentas. And this molecule binds to ligands on the decidua, binds to leptin, and helps invasion. And interestingly, in the human-specific disease, preeclampsia, the number one gene in which the expression goes up is this one, which is Siglec-6. So these are just some examples of the kinds of interesting things we're finding with regard to sialic acid biology and human evolution. Oops, what's that? Sorry, I touched something here. So I'll leave you with the thought that studying uniquely human diseases could be just as valuable as One Health and comparing humans with other animals for diseases. Thank you.